Hello my dear friends, welcome to LivePrep. In the previous lecture, we studied about the mitosis process. In that specifically, we studied about the interphase part. So in the interphase, we studied about G1 phase, S phase, G2 phase. All these things we have studied, and now we are moving to the next one, that is the M phase. But before directly going with the M phase, let us quickly recollect the information we have studied in the previous lecture, because when we are drawing the diagrams of this G1, S and G2 phases, every time we need to think about the ploidy and DNA content, what is happening to that value. And also in the diagram also, mainly we are focusing with respect to DNA, chromosome, chromatin chromosome conversions, is nuclear membrane present or absent, where the centrosomes are present, what is happening to that. These are the main focus in our diagrams. Okay, so with respect to that we are studying, let us quickly draw this. In the case of G1 phase, nuclear membrane is present, single thread-like structures are present, like this 46 are present in human. All our diagrams are with respect to human diploid cell, right? So with that only we are starting. So it is like this, and a single centrosome is present here. So here it is a 2N and a 2C. This is what we have studied. And in the case of S phase, what we have studied, nuclear membrane is present and this DNA is undergoing duplication. So because of this chromosomal duplication occurred and sister chromatids are produced, and like this how many are present? 46 are present. And not only this, in the cytoplasm centrosome duplication also occurred. And here what is the overall value here? Ploidy remains the same, but the DNA amount or DNA content is getting doubled. So it will become 4C. And the same diagram even for G2 also. For G2 also we have drawn the diagram like this one. So this is what we have studied, and now we are proceeding with the next one, that is the M phase. So this is the M phase. M phase means it is the mitotic phase. It is the mitotic phase because this is where actually the cell division is occurring. So this is the actual division phase. This is the actual division phase. This is the actual division phase. And you can also call this as cell division proper. Already we studied all these names. Let us quickly revise this. This is also called as cell division proper. Cell division proper. And regarding the timings, what we have studied, it will take only just 1 hour out of 24 hours in the case of human cell. Yes or no? And next one, with respect to percentage, if you are talking, it is taking just only less than 5% duration. That means quickly it is getting completed. Clear? This is what the information is. Next one, if you are talking about this 1 hour duration like this, so for the total we are taking 24 hours. Out of this, G1 is taking approximately 11 hours, S is taking approximately 8 hours, and G2 is taking approximately 4 hours. 11 plus 8, 19 plus 4, 23. Yes or no? And M phase is taking approximately 1 hour. This is what the approximate durations of these particular phases. So now we are talking about the M phase. So in M phase what happens? This particular, it is taking just only 1 hour. This is what we have studied. Yes or no? And regarding the G1 phase, what we studied with respect to organellar duplication. In the case of your G1 phase, most of the organelles are undergoing duplication. This is what we studied. Regarding the S phase, what we studied, centrosome duplication we have studied. And in the G2 phase actually, though not given in NCERT, if you can remember. In the G2 phase actually, mitochondria and plastids will divide. So plastid and mitochondrial duplications are the divisions. Actually, they will divide by fission, right? Mitochondria and plastids, they are almost like bacteria, and they are dividing by the fission. So that will mainly occurs in the G2 phase. If you can remember. That's it. So with this, these divisions are all completed. Now we are focusing on this M phase. This is what the basic information for the M phase. Clear? And now in NCERT, they used one particular sentence. So here, this is the most dramatic stage. This is the M phase is the most dramatic phase. Why it is the most dramatic phase? Because reorganization of almost all components are taking place. In the case of M phase, what is happening? Reorganization of almost all the types of components are taking place. What exactly happening here? Let us begin with this. Before the M phase, before starting of the M phase, what happens? Nuclear membrane is present. Chromatin means thread-like structures are present. Yes or no? And then endoplasmic reticulum is present. Golgi complex is present. Nucleolus is present. Nuclear membrane is present. These many are present when before the start of M phase. And again at the end of M phase, all these are present. Again at the end of M phase, all these are present. But in between what happens? In between what happens? Nuclear membrane disintegrates, endoplasmic reticulum disintegrates, Golgi complex disintegrates, nucleolus disappears, and then this chromatin will undergo condensation. So in this way, before and after are almost same only, but in between they are undergoing so many changes. That means in the entire M phase, if you are observing, it is a most dramatic period of the cell cycle process, where almost reorganization of almost all the types of components is taking place. So that's what the another sentence that they have added. But anyhow, already we studied that it is called equational division because the ploidy of parent cell and the ploidy of the daughter cells are same, and it is called as somatic cell division because somatic cells divide by mitosis. Understood or not? But mitosis occurs in both somatic cells and germ cells. Please remember this point. This is what some basic information that they have given. And now we are going with some specific process. M phase is divided into two subphases. Already we studied this. One is called nuclear division. Nuclear division is also called as karyokinesis, and the other one is called cytoplasmic division, that is cytokinesis. Cytoplasmic division is also called as cytokinesis. And within this karyokinesis, nuclear division, again four phases are there. They are prophase, metaphase, anaphase, and telophase. Yes or no? And these are in the sequence. If there is a fixed sequence, already there is a separate lecture that was uploaded under the introductory information. In the introductory information, faculty introduction was there, and how to understand NCERT lines. There is a video on it, and then what to refer for NEET examination. There is a video. There are 3 videos. Out of this, how to understand NCERT lines. So in that video, I have covered a content that is related to followed by follows, preceded by precedes. How to use these particular words very clearly I have explained there, so I will not repeat that explanation again. So here in the NCERT they said some sentence like so and so phase follows, so and so is followed by so and so, likewise they have used. So please, if you have not gone through that video, so please go through that video and then come back to this, then only you can understand the NCERT words. I don't want to repeat that, already we have completed that portion. Clear? Now let us continue further. So these are the phases. Now in this lecture, first we will focus on these 3. Okay. In this lecture, let us try to understand prophase, metaphase, anaphase. The content is very much clearly given in the NCERT. And if you are following the diagram, and according to the diagram, if you are able to represent this, you can cover all the sentences, and the information is very simple, very simple. Okay. So now we are going with the prophase first. Now, before going with the prophase, first you need to recollect the G2 phase. In the G2 phase, how exactly it is present? Nuclear membrane is present, both the centrosomes are present side by side, but that means still they are present here only. Next one, here they are thread-like. The DNA means this particular genetic material is present in the form of chromatin, thread-like structure, and for each chromosome, again 2 sister chromatids are present. Both of them are attached to the single centromere. Likewise, how many are present? 46 are present individually. They are not attached to each other. 46 individually. Individually means anyhow they are distributed, like because they are tangled, right? In the case of interphase, what happens? They are distributed in the entire nucleus. So they are interconnected like this. But they are not attached, they are not paired. This is what the point is. They are not paired. They are distributed randomly so that they are appearing like a big mesh-like structure. That's it. Like that only they are appearing, but originally they are not paired in the case of interphase. And anyhow, in the mitosis, there is no pairing at all, so don't worry. So now we are going with this prophase first. So what is this prophase here? In the prophase is the first phase, right? Prophase is the first phase of the karyokinesis. But please remember, in the NCERT, before starting this, they have given one disclaimer. What is that? These are all continuous things. Prophase, metaphase, anaphase, telophase, all these are continuous things. So that's why they are saying that we cannot draw clear-cut lines here. You cannot say that, yes, this is where prophase completed. Yes, this is where metaphase completed. Sometimes these two will be overlapped because for our convenience, we are dividing that into 4 parts. But originally, it is not. After prophase, it will stop. After metaphase, it is not like this. It will not stop. It is a continuous process. For our understanding purpose, we are classifying this. That's why when you are observing different books, there may be some conflict. So they may say that this is the prophase part, this is the metaphase part, like this. But for our level, whatever they have given NCERT, that is final for us. We are following the NCERT. That's it. Okay. What happens in the prophase? Observe the diagram. Everything you can understand from the diagram itself. You don't need to separately worry about the sentences. Okay. Now this time what I will do is, once we are completing prophase, I will read the content of prophase. Once we are completing metaphase, I will read the content of metaphase. That will be easy for you. Okay. Here entire information we are studying based on the prophase. But in the NCERT, what they did is they made two diagrams. They have made one as early prophase, and other one is late prophase. Okay. But please remember, before we are starting with this, you need to know the spelling of this prophase, P-H-A-S-E, phase, prophase. There is a word that is prophase, P-R-O-P-H-A-G-E, prophase. Now what we are saying? Prophase, P-R-O-P-H-A-S-E, prophase. Is there any difference between these two? Regarding these things, please remember what to focus, what not to focus. These things also I told you in that introductory lectures, right? Please go through that. Prophase, prophase, are these two same? Are these two same? Here S is there, here G is there, but remaining spelling is same. Are they same or different? They are different. Please understand. Prophase is the first phase of karyokinesis. But what it is prophase? Phase, P-H-A-G-E, phase is the virus. Prophase is the virus. It is related to the virus. Please remember, these two are not same. These two are completely different. So spelling is very, very important. What was the spelling given in the option? You need to see that very clearly, or else you may make the mistakes. Okay. So we are talking about the prophase, early prophase, late prophase. Clear? So what exactly happens in the G2 phase? Nuclear membrane is there, but here what happens? Nuclear membrane breakdown will start here. So here it came to almost end. So we need to decrease it. Like this we can write. Okay. So nuclear membrane breakdown is starting here. Nuclear membrane breakdown is starting in the early prophase. And by the time prophase is completed, what happens? Nuclear membrane will be completely removed. It will be completely removed. That means it will undergo complete breakdown. This is the first important point you need to understand. First one is with respect to nuclear membrane. Observe point by point how exactly you are changing the diagram. Follow this. So nuclear membrane will start undergoing breakdown, and it will be breakdown will be completed and it will be disappeared by the end of the prophase. So that means once nuclear membrane is disappeared means prophase completed. Next phase, metaphase will start. That is what the logic is. Clear? Not only for nuclear membrane, endoplasmic reticulum, Golgi complex also. Because these two are reticular structures. Now recollect the content from cell chapter. Endoplasmic reticulum is a network. Golgi complex are dense reticular structures observed near the nucleus. This is our sentence we studied. So they are also reticular. What is the problem with this? Now first understand why they should be removed. Why they should undergo breakdown and removal. So nuclear membrane, endoplasmic reticulum, Golgi complex. What is the problem with them? Now understand the logic here. So what is the logic here? Inside the nucleus, this chromatin or chromosomes are present, right? In the S phase, what happened? They have become doubled. Doubled means what? Duplicated. Number remains same, but sister chromatids were formed. Now ultimately what should happen? Sister chromatids should be separated and they should go to opposite sides of the cell. So that around them a nuclear membrane will be formed, so that they are going to daughter nuclei. Later on cell will divide so that they will go into daughter cells. This should happen. Now we will see what is the problem. Suppose here there is a nuclear membrane. If there is a nuclear membrane, how chromosome will move to opposite poles? Nuclear membrane will stop it, right? So that's why nuclear membrane should undergo breakdown. You need to understand the logic also. Why nuclear membrane is undergoing breakdown? If it is present as it is, chromosomes cannot go to opposite sides. That's why it should undergo breakdown. Okay, nuclear membrane undergoing breakdown, we understood. But what is the problem with ER and GC? Now understand this. ER and GC are reticular structures, network-like structures. They are distributed in the entire cytoplasm. If this much of large network is present, how chromosomes are separated? They will stop the separation. That is the reason why ER and GC also undergo breakdown. So that's why these large ones, those which can stop the movement of chromosomes, they will undergo breakdown. So nuclear membrane, endoplasmic reticulum, Golgi complex, these are the three things that are undergoing breakdown, so that they will create some space here. They will create some easy space here so that chromosomes can easily move to opposite sides. This is what the first and foremost important logic. Okay, this is what. Next one we need to talk is with respect to chromatin. See, in the interface, whether it is G1, S or G2, whatever it is, we have drawn thread-like structures. What happens to that chromatin? Chromatin will start undergoing condensation. What happens in this condensation? Length will decrease. And till now, how they are present? In the case of up to G2 phase, how they are present? This is the plasma membrane, this is the nuclear membrane. Threads are present like this, right? Double. You need to attach. Don't forget this. Like this, they are present. Like this, they are present. So in this, all the 46 are distributed in this. And since all these are thread-like, they got tangled. When they are tangled? In the interface. In the interface, all these chromatin threads are tangled. They are like this. So they are like a big mess-like structure. And now what happens? Now it will undergo condensation. When the length decreases, what happens? Now their length will start decreasing now. Their length will start decreasing now. Like this, their length is start decreasing now. So if you are like this, total how many? Total number, don't forget, 46, okay? Same way here you can draw like this. You can draw like this. Now because of this, what happens? In the interface, when they are thread-like, they got tangled. They are making a big mess-like structure. But here what happens? Condensation is starting. Condensation is keep on happening. Please remember, maximum condensation in the next phase, metaphase. So here also you can see like this, but in the early phase, you can start visualizing. How you are able to start visualizing? Observe this content. What is the term we used for chromatin? In the interface, chromatin is indistinct. From the cell chapter, you need to recollect all this. That's the reason why I told all the points there. Interface, chromatin is indistinct. What is the meaning of indistinct? Not clearly visible. Not clearly visible because it is spreaded. It is not clearly visible. But here what happens? Condensation occurring. So what happens? Now here you can start visualizing under microscope. So chromatin you cannot see easily, but these chromosomes, now they are undergoing condensation to form chromosomes. So now you can start seeing them under microscope. This is what they are saying. This is what the point. This is what the interlinking you want. This is what I am expecting from you. This is how you just understand what happens step by step, so that it will become easy for you. Okay, this is what happening. This is what happening to chromosomes. This point is over. Nuclear membrane chromosome part is over. And next point we need to focus is centrosomes. In the G2 phase, both the centrosomes are side by side. Now here what happens? Now centrosomes are moving to opposite sides. Now it is moving like this. This is here. So now what happens ultimately? Say I am keeping one pole here. So one centrosome here and one centrosome here. Understood or not? In the prophase, early prophase, they are start moving to opposite sides. Okay, suppose initially you are drawing here, you need to write here, but in our diagrams we are writing top and bottom, so I am keeping like this. So they are moving to opposite sides. Once they are moving to opposite sides, that is beginning here. In the late prophase, what happens? They have reached opposite poles, and that too from these poles microtubules have started. I told about this particular one. Yes or no? When we studied about the centrosome and centrioles in cell chapter, that time we have studied about this. Spindle apparatus, spindle poles, all the things we studied, no? That's what. So from this centrosome, spindle fibers are coming out. Okay? Microtubules, spindle fibers are nothing but microtubules. And here we are using the two names. What are these two names? Some fibers are short. Some fibers are short fibers and they are called as astral rays. Astral rays. Whereas some fibers are long. And these long fibers are called as spindle fibers. These are spindle fibers. Please observe these names. Astral rays, spindle fibers, both of them are coming from the same centrosomes only. But shorter ones are called as astral rays and the longer ones are called as spindle fibers. So that you can easily understand the NCERT content. Okay, this is what. And next one, spindle fibers are formed in the prophase itself. But please understand, still they are not attached to chromosome. This is the most important thing. Many students are thinking that spindle fibers will attach here. No, when spindle fibers will attach to chromosome, in the next phase, metaphase, in the metaphase they will attach, not here. Okay, in this way they have given the diagram. Understood or not? So let us quickly recollect all the points. Nuclear membrane, endoplasmic reticulum, Golgi complex will undergo breakdown. Next one, inside the nucleus there is a nucleolus that will also disappear. Next one, what happens to chromatin? It will undergo condensation so that you can start visualizing them under the microscope. And another advantage is, in the interphase they are tangled, and in the prophase what happens? They will become untangled. Untangled means they are separated from each other. Now you can see them as a clear-cut structures. So in this way untangling will occur. So these are some important points. Next point is, centrosomes will start moving to opposite sides and they are forming the microtubules, and some are called astral rays, some are called as spindle fibers. Actually these spindle fibers are important in attachment to chromosomes in the next stage, metaphase. Clear? And here at this point, if you see, this is the centrosome with these microtubules. All these centrosome with microtubules. All these are, this is called as one aster. Already this information clearly I have explained in the cell chapter itself, under the centrosome itself. So here I don't want to spend more time. So please observe this. So this is like appearing like a star shaped, right? Star. So this is there the center centrosome, and from this microtubules are coming out appearing as a star. That's why it is called aster. And this is also called aster. Yes or no? So in this way what happens? Centrosome with this microtubules is called aster. This is one aster, this is one aster. That's why it is called as amphiastral. In the case of animal cell, it is amphiastral. If you are still having any doubt, please go through centrosome and centriole lecture from the Cell, the Unit of Life. There I have clearly explained all these things. Okay. Next one, plant cell. For the plant cell, let us see. So can you use the term aster for the plant cell? No, because if you are using the word aster, centrosome should be present at the center. But in the case of higher plant cell, centrosome is not present. That's why in the case of plant cell, we are saying that it is anastral. Yes or no? This is what the word we are using. Next one is, in the case of animal cell, centrosome is present. That's why it is called centric. Centric spindle poles are formed. Centric term. Okay. But in the case of plant cell, centrosomes are not present. That's why it is called eccentric. That means centrosomes are not present. Please understand these words. This is how they are using this. This amphiastral is also simply called as astral. You can say simply astral means asters are present. How many asters? 2 asters are present. That's why amphiastral. On both the sides asters are present. In this way some names we are using. But the term spindle pole, yes, spindle pole term is used here also. Spindle pole term is used for animal cell and a spindle pole term for plant cell. Here also it is used. In both the cases, all these points already we have discussed. That's why I'm not repeating this. So spindle pole term you can use in both the cases. But the thing is, in animal cell, who will form the spindle pole? Centrosome itself. Centrosome itself is helping in formation of the spindle pole, and that too from where these spindle fibers are coming out. So around the centrosome, already we studied, no? Around the centrosome, pericentriolar material is there, and there some group of proteins called satellites are present, and from there actually this spindle fibers are coming out. And what actually spindle fibers are made up of? Microtubules. Spindle fibers are made up of microtubules. In the previous lecture we studied, no, in the G2 phase, tubulin protein is produced and that tubulin is used here. That will be assembled here so that spindle fibers will be arranged here. That's it. This is what the point is. But in the case of plant cell, centrosome is not there. Now who will form the spindle pole? Already we have discussed this. Some proteins are present in the cytoplasm. So these proteins will come together, and in the plant cell, these proteins will help as spindle poles, and from there spindle fibers will arise. This is what the point already we studied. So in this way, spindle pole term is used in both plant and animal cells, but the aster term is used in animal cells. Is it clear? Now, after completion of this, we are talking about the spindle apparatus. Already this calculation also we have studied. What is a one spindle apparatus? One spindle apparatus. One spindle apparatus is also called as one mitotic apparatus. One mitotic apparatus. One mitotic apparatus is equal to two spindle poles plus spindle fibers. Spindle fibers. This is how technically you are writing, but in this particular chapter what they have given is, instead of giving two spindle poles, they have given two asters plus, instead of spindle poles they gave simply microtubules from the asters, microtubules from asters. Please understand this. In this chapter they used mitotic apparatus term, and they are saying that two asters plus microtubules from the asters. Now this is called as mitotic apparatus, but in the cell chapter under the centrosome they used a term called spindle apparatus, and it is nothing but two spindle poles plus spindle fibers. Understood or not? This is what the information is. In this way, come to this diagram. Here, how many spindle apparatus are present? In one mitosis, only one spindle apparatus is produced. What is spindle apparatus? Both the spindle poles plus all the spindle fibers. Combined, it is called as spindle apparatus. Why it is spindle? Because here if you are observing, it is having the spindle shape. So this is what the shape that you are observing here. Yes or no? That is the spindle shape. So that's why it is called a spindle apparatus. So from here to here, if I am saying that center of spindle apparatus means here it will come. And that word we will use in the next phase, that is the metaphase. Okay. So this is some general basic information that you need to understand. So at the end of this prophase, what happens? This all will disappear. That's it. With this prophase will be completed. That's it. But we need to talk about ploidy and DNA content also. In the G2 phase, what is the ploidy? 2n. And what is the C value? It is 4C. And here you please tell me. Here also chromosomes are present, the same number remains same only, just condensation occurred, that's it. There is no change in the number. So here also it is 2n only. But what, and DNA content also, just DNA condensed, that's it. There is no change here. So that's why it will be 4C. So ploidy and DNA content is same like G2 phase only. So this is what the information that we need to understand with respect to prophase. I hope now you understood this. Simple information. Try to follow the diagram. So you should be in a position to draw this diagram. So once you know how to draw the diagram, then based on that you can write the entire content. This is what the information that you need to understand. Clear? So with this particular one is completed. So now let us go through this. So M phase, general information regarding the M phase they give. This is the most dramatic period of the cell cycle, involving a major reorganization of virtually all components of the cell. In the M phase, what happens? Initially all of them are present, and then they are undergoing disappearance, breakdown and all the things. Again at the end, again they are reappearing again. So in this way, virtually for all the components there are some changes, reorganization is taking place. That's why they are saying that this is the most dramatic period. Next one. Since the number of chromosomes in the parent and progeny cells is same. So ploidy of the parent cell and the ploidy of the daughter cell. That means number of chromosomes is same. It is also called as equational division. Next one. Though for convenience, mitosis has been divided into 4 stages of nuclear division. Within the nuclear division, karyokinesis, 4 stages we are studying. Even though we are dividing into 4, it is very essential to understand that cell division is a progressive process. It is a continuous process. It is not stopping in between. It is a continuous process, and very clear-cut lines cannot be drawn. We cannot clearly say that this is the end of prophase, this is the end of metaphase. Like that we cannot say. Okay, next one. Karyokinesis has the 4 stages. Sequence is fixed: prophase, metaphase, anaphase and telophase. And first we are going with the prophase. Prophase, which is the first phase of karyokinesis of mitosis, it follows. I told you how to use the word follows and followed by. If they are saying about only one phase, then what you need to write? So this prophase follows G2 phase. This is the correct. But here they have given before G2 also. So G2 is following S phase. Okay, that means first S will come. After that G2, after that prophase. So G2 follows S phase. Prophase follows G2 phase. So here they are saying that G2 prophase follows S and G2. Like this they are saying. This is what the sentence uses. Observe carefully. So in this way, karyokinase, first phase of karyokinesis of mitosis, and this is following S and G2 phases. If they are giving only one, then you need to go for G2 phase only, okay, of interphase. Next one, in the S and G2 phases, the new DNA molecules formed are not distinct. What is the meaning of not distinct? S and G2 are interphase. Not distinct means not clearly visible, thread-like. They are distributed like a network. That's why that's how they are present. But intertwined means they are coiled like this, because they are randomly distributed. They are present like a mesh like structure. That's why intertwined. Next one, prophase is marked by initiation of condensation of chromosomal material. How we can understand that prophase is started? Once chromosomal condensation begins, we can understand that prophase is started. This is what the important point. Next one, the chromosomal material becomes untangled. Untangled means what? Till now they are coiled like this. Now they get separated from each other. Each chromosome is getting separated from each other. So now you can see them clearly under microscope. That's what they are trying to tell us. The chromosomal material becomes untangled during the process of chromatin condensation. So chromatin will undergo condensation, so slowly trying to become chromosome. So they are separated from each other. Untangled means separated from each other during this process. The centrosome, which had undergone duplication in S phase, when the centrosome duplicated in the S phase, there it got duplicated, and now means what? In prophase, begins to move towards opposite poles. There it duplicated, but now it is moving to opposite poles in prophase. Next one, the completion of prophase can be obtained by. So how you can say that prophase completed? There are some features are there. Based on that, we can expect that prophase completed. What are those? Chromosomal material condenses to form compact mitotic chromosomes. So condensation is keep on happening, so that after the next phase, that is the metaphase, there the maximum condense. So condensation is almost completed. So you can say mitotic chromosomes. And chromosomes are seen to be composed of two chromatids. Sister chromatids are still present, right? Still they are attached at the centromere. So sister chromatids you can see, and they are attached at the centromere. This is how you can see. Next one, centrosome, which had undergone duplication during interphase. In the interphase means where? In the S phase they have undergone duplication. Now they begins to move towards the opposite poles of the cell. Now they are going to opposite sides. Next one, each centrosome radiates out. Radiates out means gives out. From that they are coming from all the directions. What? Microtubules, and they are called as asters. Okay, so these centrosomes are giving out the microtubules. So centrosome with the microtubules is called as asters. In this way, two asters are present. Next one, the two asters together with spindle fibers, they will form the mitotic apparatus. So spindle poles plus spindle fibers is called spindle apparatus or mitotic apparatus. That's what they are saying. But technically the term aster is used for animal cell, but the spindle pole term is used in both animal and plant cell. Next one, cells at the end of prophase, when viewed under the electron microscope, when we are observing under the microscope, they do not show Golgi complex, they do not show endoplasmic reticulum, they do not show nucleolus, and they do not show nuclear envelope. That means what happens? Upon condensation, chromosomes are formed, and these chromosomes are formed directly in the cytoplasm. That is what the meaning is. Early prophase breakdown started, and late prophase breakdown almost completed, and chromosome condensation began, and now here almost it is becoming condenser rod like structure. This is what they are saying. But please remember, they are still distributed in the entire cytoplasm. They are distributed. Okay, this is what, in the case of animal cell, it is the amphiastral spindle, and here it is the centric means centrosomes are present. In the case of plant cell, anastral, asters are not present, eccentric, centrosomes are not present. Okay, so you can also call this spindle as a bipolar spindle. What is the meaning of bipolar spindle? Two poles are present for the spindle apparatus. So that's why it is called the bipolar spindle apparatus is seen in these cells. With that, prophase is completed. Now we are going with the metaphase. In the metaphase, what happens? This is the stage of our prophase. Late prophase means prophase ending, right? Yes. Now what happens in the metaphase? Simple. The spindle fibers formed in prophase, right? Now they will go and attach to chromosome. and then they will pull it to the center. Because why they are pulling? Because now they are randomly distributed in entire cytoplasm, right? Now they are randomly distributed in entire cytoplasm. Now observe the diagrams carefully. Now we are going with the metaphase, middle. Meta is the middle. So chromosomes will come to the middle. That's why it is called metaphase. Now here how you are drawing the diagram. This is the plasma membrane. Will you draw nuclear membrane here? No. At the end of the prophase 1, nuclear membrane disintegrated. That means the disappearance of nuclear membrane means metaphase started. At the end of the prophase 1, nuclear membrane disappears. That means immediately after that, metaphase will be started. This is what the point is. So now here what you need to draw? Here is one spindle pole. Here is one spindle pole. From this spindle fibers are coming out. Yes or no? From this spindle fibers are coming out. And now what happens? Initially what happens? Chromosomes are present randomly. Here and there they are present. So randomly I am drawing like this. Okay? Like this they are present. Randomly they are present in the cytoplasm. Like this how many are present? 46 are present. So now what we need to do? Observe the diagram carefully. Suppose this is the chromosome. For this chromosome what happens? One spindle fiber from one pole and other spindle fiber from other pole. Yes or no? From this one, from this one, like this, we need to attach like this. So please observe this content carefully. This is very important. Now what happens? This is the chromosome. And for this chromosome, already we studied about the centromere. Where the spindle fibers will attach? At the centromere. How they are attached? To the centromere, on one side, one kinetochore, other side, other kinetochore. Under the cell chapter we studied, no? Under the cell chapter, what we have studied? Suppose this is the centromere. For the centromere, on one side, one kinetochore, other side, other kinetochore. Likewise, for one centromere, two kinetochores. Calculation also I told you, right? For one centromere, two kinetochores are present. Now what happens? For each chromosome, one centromere are present and two kinetochores are present. And for this one we forgot, right? And here it will go and attach like this. Now observe carefully. Now here is the kinetochore. To this kinetochore, spindle fiber will attach. Here there is a kinetochore, spindle fiber attach. Kinetochore, spindle fiber attaches. Kinetochore, spindle fibers. Kinetochore, spindle fiber. Like this. Understood? Like this for all the 46 chromosomes. Initially all the chromosomes are randomly present in the cytoplasm. Now what happens? Spindle fibers from opposite poles, they will come and attach to each chromosome. Observe any chromosome. One spindle fiber from one pole, other from the other pole. Likewise, everywhere they are attached. And now you can call this as early metaphase. So please understand, in the prophase, spindle fibers are formed, but they are not attached to chromosome. When they are attached? In the metaphase only. If you are not saying early and late, simply if you are saying metaphase, in the metaphase itself, spindle fibers from opposite poles, they will come and attach to the chromosomes. That is happening. And then what happens? And then if you are calling this as a late metaphase, now what happens? Spindle poles are at the same location only. Spindle poles are present at the same location only. But what happens to these chromosomes? All the chromosomes will come to the center. Now I will give you two options. First option is center of the cell. Second one, center of spindle apparatus. Center of spindle apparatus. Two options I am giving you. Tell me which one is the correct one. In the NCRT, in one sentence they used center of the cell. In another sentence they used center of the spindle apparatus. I am only telling you. In NCRT they used both the sentences, but if you are talking technically, only one is correct. Which one is correct? Tell me. Which one is correct? If you are technically talking about this, now you are saying that chromosomes will come to the center. But center of what? Center of spindle apparatus, not to the center of the cell. And the thing here is, in majority of the cases, center of spindle apparatus is center of the cell. That's why you are saying center of the cell. But in some cases, rare cases, center of spindle apparatus is not the center of the cell. Please understand this. But it is always center of the spindle apparatus. That's why in general if they say okay both of them are correct, but if they are asking these two as two separate options and you need to select only one, then which one you need to select? Center of the spindle apparatus you need to select. Please understand this. Many students are making mistake here also, and unnecessarily they are arguing that in the NCERT itself they gave that it is center of the cell. In the same NCERT separately they gave it is center of spindle apparatus. Now which one you will consider? You need to consider center of the spindle apparatus. Please observe this carefully. Clear? Now what you need to do? Now spindle apparatus, that means from this pole to this pole, this portion is the spindle apparatus. For the spindle apparatus center, let us take this is the center. Now in this diagram, this is the center for cell also. In this way we are drawing the diagram. Now I will tell you, suppose in some cases what happens is, suppose here is one pole, here is one pole, exceptions are there. Now what happens? This will be the center of spindle apparatus. Yes or no? This will be the center of spindle apparatus. Is it center of the cell? No. So that's why which one is more correct? Center of spindle apparatus is more correct. But anyhow, we are drawing the diagram for normal condition. In the normal condition, here is the spindle pole. So center of spindle apparatus is nothing but center of spindle. Center of spindle apparatus is nothing but center of the cell. Now let us draw the diagram. Now how we are changing the diagram? All the 46. Please remember there are no pairs. This is one chromosome. This is another chromosome. This is another chromosome. Likewise, all the 46 chromosomes we are writing at the center. And how they are coming here? Please observe this thing. Here is one kinetochore. Here is one kinetochore. For each centromere, there are two kinetochores like this. And how the spindle fibers are attached? Spindle fibers from the opposite poles are attached to each chromosome, so that all the chromosomes are aligned at the center of the spindle apparatus. Initially they are randomly present, spindle fibers are going and attaching to them, and now they are bringing all the chromosomes to the center of spindle apparatus. So in this way they are coming here. That's it. Metaphase completed. Metaphase completed. That's it. So here what are the points? Nuclear membrane completely absent. And next one, chromosomes are maximum condensed, means fully condensed, maximum condensed. This is the maximum condensation. So that's why what happens? You can clearly see chromosome morphology. Chromosome morphology is clearly studied. Chromosome morphology means it is the chromosome structure, not the shape. Please observe this. Chromosome structure. Chromosome structure is clearly seen. Chromosome morphology means chromosome structure is clearly seen in the metaphase. That is the reason why for majority of our experiments we are isolating chromosomes when they are present in the metaphase. Please observe this. That's why metaphase chromosomes we are isolating, because maximum condensed and clearly visible, and you can easily study the structure and many things. That's why this is the most important thing. And next point is, and these chromosomes, to these chromosomes, spindle fibers from the opposite poles are attached in such a way that to each chromosome, spindle fibers from opposite poles are attached. Where they are attached? To the kinetochores. And what are these kinetochores? They are also a type of proteins. And where these kinetochores are present? They are attached to the centromere. Understood or not? So indirectly what we are saying? Spindle fibers from opposite poles will attach to centromere through kinetochore. This is how they are arranged. That too, here there are no pairs. Individually all the chromosomes are present side by side. Side by side all the chromosomes are arranged. Next one, for the kinetochores if they ask, one chromosome has two kinetochores. In the kinetochores, one kinetochore will attach to one spindle pole and other kinetochore will attach to other spindle pole of their own sides. Their own side means this side kinetochore is attached to this side pole and that side kinetochore is attached to that side pole. Same is the condition for all the chromosomes. That's it. This is how they are present in the metaphase. And now technically if you are observing, for all these chromosomes, the centromere is present in one line, so that it is appearing as a plate or the plane at all are present in one plane, and this is called as metaphase plate. It is called as metaphase plate, and you can also call this as equatorial plate. Equatorial plate. Equatorial plate. Understood or not? So, in the case of metaphase, all the chromosomes will align at the equatorial plate or the metaphase plate. This is what they are saying. That's it. With this, metaphase is completed. Now we will directly go over the ploidy and the DNA content. How many chromosomes are present? 46 only. So chromosome number remains same. It is 2n only. What is the DNA content? That is 4c only, as it is, right? Simply arrangement is different. That's it. There is no change in number of chromosomes. There is no change in DNA content. It is same only. So this is what about the 2n 4c. That's it. Completed. This is what about the metaphase. And now let us quickly go with the NCERT sentence. That's it. Diagram wise observe it and then read the sentences. You will understand every point. The metaphase. The complete disintegration of nuclear envelope marks the start of second phase of the mitosis. That means at the end of the prophase, nuclear membrane disappears. Once that is disappeared means metaphase will be started. So this is what they are saying. Hence, the chromosomes are spread through the cytoplasm of the cell. Since nuclear membrane is not there, where the chromosomes are present? In the cytoplasm. Now recollect the sentence from cell chapter. During different stages of cell cycle, structured chromosomes are formed in place of nucleus. In place of nucleus means where? In the cytoplasm these are present. This is what the sentence is. Interrelate the content. Next one. By this stage, condensation of chromosomes is complete. That means maximum condensation you can see in metaphase of mitosis. This is what they are saying. And they can be observed clearly under the microscope. That's why distinct term you are using. And then in the answer, in the nucleus part of the cell chapter, they used structured chromosomes. Why they are saying structured chromosomes? Because chromatin is indistinct, not clearly visible. But these are clearly visible. That's what they are saying. Next one. This then is the stage at which morphology of the chromosomes is most easily studied. Morphology means chromosome structure is studied. Shape, anaphase. Chromosome shape means whether V shape, L shape, J shape, I shape. That shape we will study in the anaphase easily. But the morphology means chromosome structure. Better underline the chromosome morphology and write chromosome structure is easily studied in the metaphase. At this stage, metaphase chromosome is made up of two sister chromatids. Observe the diagram. Observe the diagram. Each chromatid has each chromosome has two chromatids, sister chromatids, because both of them are still attached to same centromere. So each chromosome has two sister chromatids. Next one, which are held together by centromere. Still they are attached at the same centromere. Next one, small disc-shaped structures at the surface of centromere are called as kinetochores. There are two disc-shaped structures called kinetochores are present for each chromosome. Next, these structures serve as the sites of attachment of kinetochores. That means these sites of attachment for the kinetochore. These structures are acting as sites of attachment of spindle fibers. So spindle fibers from the opposite poles will come and they will attach to these kinetochores. So this is how they are present. Next, these spindle fiber, this kinetochore they are saying formed by the spindle fibers. So here they are saying that so kinetochores are also having microtubules formed by spindle fibers. In this sentence meaning is they are also having microtubule, but please remember kinetochores technically they are having different other types of proteins, but here they gave like this, so we cannot do anything. So simply remember this point. Okay, so these structures means kinetochores. These structures means kinetochores serve as the sites of attachment of spindle fiber. That means originally, even though you are saying that spindle fibers are attached to centromere, originally they are attached to kinetochores of centromere. Okay, to the chromosomes, and that are moved into the position. They are moved into the position at the center of the cell. So in this way they are pulling this. The spindle fibers are pulling all these chromosomes to the center of the cell. Here they are saying center of the cell, but here they are saying that they are coming to the center of the spindle apparatus. I will tell you clearly. So here, even though they are saying center of the cell, what is technically more correct? Center of spindle apparatus is more correct. Hence, the metaphase is characterized by all the chromosomes coming to lie. Lie means present at the equator. That means in the metaphase, all the chromosomes are present at the equator. Okay, that is the center of the spindle apparatus, with one chromatid of each chromosome connected by its kinetochore. understand this word chromatid. Two chromatids are present, two sister chromatids, no, chromatid. One chromatid side, one kinetochore is there, other chromatid side, other kinetochore is there. So in this way, equator with one chromatid of each chromosome connected by its kinetochore to spindle fiber from one pole, and its a sister chromatid connected by its kinetochore to spindle fiber from the opposite pole. What is the meaning of this particular sentence? Each chromosome has two chromatids. One chromatid, through its kinetochore, it is connected to spindle pole of that side, and a sister chromatid through its kinetochore connected to spindle fiber coming from the opposite pole. Means this is connected to this side pole. This is what the meaning is. So everything you can understand from the diagram. So diagrams are very, very important. Okay, next one. Next one, the plane of alignment of chromosomes at the metaphase is referred to as metaphase plate. The level at which they are arranged, that is called as metaphase plate. Next one, the key features of the metaphase are, what are the important features of the metaphase? Spindle fibers attached to kinetochores of chromosome. Please remember, spindle fibers are formed in the prophase, but they are attached to chromosomes in metaphase. Next one, chromosomes are moved to the spindle equator. At the middle of that spindle apparatus, they are coming to the spindle apparatus equator, spindle apparatus equator. That means center of the spindle apparatus they are coming. Please remember, technically this is more correct. It is not the center of the cell, center of the spindle apparatus is more correct. And get aligned along the metaphase plate through spindle fibers to both the poles. So spindle fibers from both the poles are coming, so that all the chromosomes are present at the center. That center is called as metaphase plate. So in this way all the chromosomes are present, and in this way initially randomly they are present in cytoplasm, but now all of them will come to the center of the spindle apparatus. This is what they are saying. So in this way, here they are saying that chromosome morphology can be easily studied. So in this way easily we can isolate the chromosomes. So in this way we can prepare this particular one, and this type of representation of chromosomes is called as karyotype. Please remember this word. This was not mentioned here, but in class 12th there is a chapter called Principles of Inheritance and Variation, and in that chapter we will study about this karyotype word. So please remember this. So metaphase chromosomes are important, and metaphase chromosomes are isolated, and then karyotype is prepared, and based on the karyotype any abnormalities that are present in the chromosomes can be easily studied. This is what the point is. Okay, better we will start it here because anaphase will take little bit more time, and this we can cover in the next lecture. Okay, so with this this lecture will be completed. So in the next lecture we will go with the remaining phases of the cell cycle. Okay, thank you so much my dear friends. We will meet in the next lecture. Thank you.